So the next we have is enzymes as targets of drug action. Enzymes are basically the potential site for drugs because they play a very crucial role in many biochemical pathways and processes in the body. Let's just discuss what they are. They are the proteins that catalyze specific chemical reactions, and the drugs that target enzyme can either inhibit it, which by the way I have shown by these cross lines, or they can either enhance their activity, which I have shown by these green tick lines. So the inhibition of enzyme is a common strategy in drug development, as it can lead to suppression of a particular biochemical pathway or process that may be contributing to a disease. Let's just take statins for example. Statins inhibit HMG coenzyme reductase, which is responsible for the synthesis of cholesterol in the liver. It is used in hyperlipidemia, basically to reduce LDL. LDL is more dangerous as compared to the HDL. LDL is abbreviated as low density lipoproteins, HDL high density lipoproteins. So what it does is, it basically inhibits the HMG coenzyme reductase, which in turn reduces the cholesterol. On the other hand, enhancing the activity of certain enzymes can also be therapeutic. For example, some drugs target enzymes involved in neurotransmitter synthesis or degradation, which can be used to treat psychotic disorders like depression, anxiety. So here I have given an example, which is donepezil. Donepezil is a medication used to treat the symptoms of Alzheimer's disease, which is a progressive neurodegenerative disorder that affects cognitive function. So donepezil enhances activity of acetylcholine in the brain by acting on acetylcholinesterase enzyme. What it does is, it is responsible for breaking down the neurotransmitter acetylcholine in the brain. So by inhibiting acetylcholinesterase, donepezil increases the concentration of acetylcholine in the brain, which can help improve cognitive functions in the patients with Alzheimer's disease. So the third example we have is monoamine oxidase inhibitors, which are a class of drugs that target enzymes involved in neurotransmitter degradation, like the previous one we have discussed before. So MAO inhibitors, what they really do is inhibit the activity of monoamine oxidase, an enzyme responsible for breaking down the neurotransmitters such as serotonin, dopamine, norepinephrine. And by inhibiting these enzymes, MAO increase the availability of these neurotransmitters in the brain, leading to improved mood and other therapeutic effects. So we have discussed two inhibitory protocol examples, like statins inhibit HMG coenzyme reductase and they reduce the cholesterol, while the monoamine oxidase inhibitors inhibit the monoamine oxidase and they increase the serotonin, dopamine, and these all neurotransmitter molecules. And the other we have discussed is about donepezil, which enhances the effects of an enzyme, acetylcholinesterase. So this is all about enzymes. Please take snapshot of it for future remembering, or you can also access the PDFs. Next we have is receptors as targets of drug action. In the previous slide, we have discussed about enzymes as targets of drug action. Now we will discuss about receptors as targets of drug action. So basically they are the potential site of action for drugs. Let's just discuss what are receptors. So receptors are transmembrane proteins on the cell membrane, and they are responsible for mediating the effects of many signaling molecules in the body, including like neurotransmitters, hormones, growth factors. In short, drugs can interact with them in a variety of ways to modulate their activity. One way that drugs can target receptors is by binding to the receptors and mimicking the effects of an endogenous ligand. For example, opioids. Opioid drugs like morphine and fentanyl bind to the opioid receptors in the brain, mimicking the effects of endogenous opioids like endorphins and enkephalins. So what they do is they trick the brain into believing that these are endorphins and enkephalins, and in turn they reduce the pain relief, sedation, and other therapeutic effects. Next example, we have propranolol. Alternatively, drugs can also target receptors by binding to them and blocking the effects of an endogenous ligand. For example, beta blocker like propranolol we have discussed here bind to the beta-adrenergic receptors in the heart, blocking the effects of adrenaline and reducing the heart rate and blood pressure in return. This can be useful in treatment of hypertension and other cardiovascular conditions. So these are receptors as targets of drug action. Next we have is ion channels as targets of drug action. So the ion channels are another important class of drug targets. And basically these are membrane proteins that form channels in the cell membrane, allowing the selective passage of ions such as sodium, calcium, potassium, and chlorides, obviously, across the membrane. Drugs that target ion channels can modulate their activity in a variety of ways. For example, drugs that block ion channels can prevent the movement of ions across the membrane, leading to changes in the membrane potential and inhibiting cellular function. One example of such drug is lidocaine. So what lidocaine does, it basically blocks sodium channels and is used as a local anesthetic. It blocks the sodium channels and the nerve conduction is inhibited. As a result of it, numbness in the local area is seen. So conversely, drugs that activate ion channels can enhance their activity, leading to increased ion influx across the membrane and the activation of downstream signaling pathways. For example, drug like nicotine, which activates nicotinic acetylcholine receptors and can lead to increased dopamine release and a sense of pleasure and reward. So nicotine, as you know, is present in the cigarette. So what it really does is it binds to the nicotinic acetylcholine receptors and dopamine is released. Now, ion channels are also an important target for the development of drugs for neurologic and cardiovascular disorders. For example, drugs that target voltage-gated calcium channels can also be used to treat hypertension, while drugs that target voltage-gated sodium channels can also be used to treat epilepsy and other seizure disorders. So carbamazepine, it is used as an anti-seizure drug. So what it really does, it blocks the nerve conduction via sodium channel blockage, and as a result of it, anti-seizure properties are exhibited. So the calcium channel blockers act by blocking the influx of calcium into the vascular smooth muscles and cardiac muscle cells during membrane depolarization. Because the muscle contraction is largely dependent upon influx of calcium, its inhibition causes relaxation, particularly in atrial beds. So this is all about ion channels as targets of drug action. Next, we will move on transporters as targets of drug action. Now, transporters are another important class of drug targets. So these are membrane proteins that transport a variety of molecules such as ions, neurotransmitters, and drugs across the cell membrane. So transporters play a very critical role in maintaining cellular homeostasis and regulating the concentration of molecules in the extracellular space. So the drugs that target transporters can modulate their activity in a variety of ways. For example, drugs that inhibit transporter activity can block the reuptake of neurotransmitters from the synaptic cleft, leading to increased level of neurotransmitters and enhanced neurotransmission. One example is SSRIs, which is selective serotonin reuptake inhibitors, which just bind to the serotonin transporter proteins in the synaptic cleft, and what they do is they block the reuptake of serotonin. So when the reuptake of serotonin is blocked, serotonin will reside there in the synaptic cleft and produce its effects, which are like reward mechanism, and the person may feel jolly. Conversely, drugs that activate transporters can also enhance their activity, leading to increased transport of molecules across the membrane. One of such example is amphetamines, which bind to the monoamine transporters, and it increases the release of neurotransmitters such as dopamine and norepinephrine. Next we have is SGL2 inhibitors, which are a class of drugs used for the treatments of type 2 diabetes. So these drugs work by blocking the action of SGL2 transporters in the kidneys, thereby reducing the reabsorption of glucose and increasing the amount of glucose excreted in the urine. This results in lower blood glucose levels and can help improve glycemic control in patients with type 2 diabetes. So SGL2 transporter is abbreviated as sodium glucose cotransporter 2. The SGL2 transporter is a protein found in the cells lining the kidney tubules. So that is all for transporters. Next we have is DNA and RNA as targets of drug action. So DNA and RNA are also important targets for drug action, particularly in the treatment of cancer and viral infections. So DNA, which is like this double-stranded, is a genetic material that carries the instructions for the development and function of like all living organisms, while the RNA, which is single-stranded, is a molecule that plays a critical role in the protein synthesis and gene expression. Now, the drugs that target DNA and RNA can modulate their activity in a variety of ways. For example, drugs that inhibit DNA synthesis can prevent the replication of cancer cells, leading to cell death and tumor regression. One example of such a drug is cisplatin, which binds to the DNA and inhibits DNA replication and is used to treat various types of cancer. Now, similarly, drugs that inhibit RNA synthesis can prevent the replication of RNA viruses, leading to decreased viral load and improved clinical outcomes. One example of such drug is remdesivir, which inhibits the viral RNA synthesis and has been shown to be effective in the treatment of COVID-19. So what it does is, it binds to the viral RNA-dependent RNA polymerase and inhibits viral replication by terminating the RNA transcription prematurely. Now, how remdesivir really does that? Once inside the body, remdesivir is converted to analog of adenosine triphosphate, a molecule that is essential for the cellular energy metabolism. The active form of remdesivir is then incorporated into the replicating RNA strand of the virus, which interferes with the normal replication process and results in premature termination of the RNA chain. This disrupts the ability of virus to replicate and reduces the amount of virus in the body. Now, this is RNA and DNA as targets of drug action. Next, we will move on towards the metabolic pathways as targets of drug action. So the metabolic pathways are complex biochemical processes that occur within the cells to convert nutrients into energy and building blocks for cellular processes. Now, these pathways involve a series of enzymatic reactions that are tightly regulated and coordinated to maintain cellular homeostasis. Now, first we will take an example of methotrexate, but before that, let us just understand how folic acid is converted into DNA and RNA. So, folic acid is converted into dihydrofolate via dihydrofolate reductase. Then dihydrofolate is converted into tetrahydrofolate via the same enzyme, and then the tetrahydrofolate is converted into thymidyllic acid, which is useful for the DNA, amino acid synthesis, and purines, which are the important step for the DNA and RNA. So tetrahydrofolate can conserve back to dihydrofolate via an enzyme thymidylate synthase. Now, what methotrexate does is, it is an anti-metabolite drug that inhibits the enzyme dihydrofolate reductase, a process that converts dihydrofolate to tetrahydrofolate, which is essential for the synthesis of purines and pyrimidines, the building blocks of DNA. Now, cancer cells are typically have a higher demand of nucleotides compared to the normal cells, and thus they are more sensitive to the effects of methotrexate. By inhibiting the dihydrofolate reductase, methotrexate prevents the formation of tetrahydrofolate, leading to a depletion of nucleotides and ultimately inhibition of DNA synthesis and cell division. This results in the death of cancer cells. Paclitaxel, on the other hand, is a chemotherapy drug that targets the microtubule assembly pathway, which is essential for the cell division. Microtubules are long cylindrical structures that form the framework of cell and play a critical role in cell division. Now, during cell division, microtubules form the mitotic spindle, which separates the duplicated chromosomes into two daughter cells. Paclitaxel works by binding to the microtubules and stabilizing them, which prevents their disassembly and interferes with the normal process of cell division. Now, this results in formation of abnormal mitotic spindles and the arrest of cell division, ultimately leading to apoptosis or the cell death. Apoptosis is the programmed cell death, necrosis is the accidental cell death. Overall, methotrexate and paclitaxel target different pathways in the cancer cells, but both disrupt the normal process of cell division, which is a hallmark of cancer. By targeting these pathways, these drugs can effectively kill cancer cells and help to treat cancer. Next we have is signal transduction pathways as targets of drug action. The proteins involved in signal transduction pathways are also important targets for drug action. Signal transduction pathways are a complex network of proteins and other molecules that are involved in transmitting signals within cells and between cells. These molecules play a critical role in a variety of cellular processes, including cell growth, differentiation, and survival. Now here, we will take an example of imetanib, which is a medication used to treat certain types of cancers such as chronic myelogenous leukemia, gastrointestinal stromal tumors, and some other rare cancers. Its mechanism of action involves targeting specific enzymes known as tyrosine kinases, which are involved in the growth and division of cancer cells. Imetanib works by binding to the ATP binding sites of these enzymes, which prevents them from functioning properly. Specifically, imetanib targets the BCR-ABL kinases. Now, these are the enzymes found in CML cells and is responsible for their uncontrolled growth and division. By inhibiting the BCR-ABL kinase, imetanib is able to reduce the number of cancer cells in the body. Now, imetanib basically binds itself to the substrate and prevents the ATP from binding to the BCR-ABL kinases. Now, imetanib also targets other tyrosine kinases that are involved in the growth and division of cancer cells, such as the c-KIT kinase found in the GISTs. Overall, imetanib's mechanism of action involves targeted inhibition of specific enzymes that play a key role in cancer cell growth and division, leading to the suppression of tumor growth and potentially even cancer remission. Now, another example we are taking here is CAMP pathway. The CAMP pathway is a cellular signaling pathway that is involved in many physiological processes, including regulation of cardiac function and bronchodilation in the lungs. Drugs that target this pathway can be used to treat heart failure and asthma. For heart failure, drugs that target the CAMP pathway include the beta-adrenergic receptor agonists such as dibutamine and isopropranolol. These drugs activate the beta-1 receptors on the surface of cardiac cells, which leads to an increase in intracellular CAMP levels. This in turn enhances the cardiac contractility and increases heart rate, improving cardiac function in patients with the heart failure. For asthma, drugs that target the CAMP pathway include beta-adrenergic receptor agonists such as albuterol and salmeterol. These drugs activate the beta-2 adrenergic receptors in the smooth muscles of the airway, which leads to an increase in intracellular CAMP levels. This causes relaxation of smooth muscles and dilation of the airway, improving breathing in patients with asthma. In addition to beta-adrenergic receptor agonists, other drugs that target the CAMP pathway can be used to treat heart failure and asthma. For example, phosphodiesterase inhibitors such as milrinone can increase CAMP levels by preventing the breakdown of CAMP by phosphodiesterase enzymes. These drugs can also improve cardiac function and bronchodilation, respectively. Now, ATP is converted into CAMP by adenylate cyclase. CAMP is converted back to adenosine monophosphate by phosphodiesterase. Now, this CAMP is important because in the heart, it acts on beta-1 receptors and produces its effects, while in the lungs, it acts on beta-2 receptors and produces bronchodilation or smooth muscle relaxation. Now, this is all about today. Keep following sqadia.com for more such videos in the future. Bye-bye.
Watch our medical videos anytime and anywhere. Download sqadia.com app now.